increase the chances of anything to do with blood vessels and cognitive impairment all the way to so-called vascular, non-Alzheimer's type dementias. So that's the importance of these early studies with ashwagandha in humans. But why couldn't they just do a study of 500 people? You know, you would require interest and funding sources to do this. And maybe you don't have to do 500 all at once, but in the ways that we are used to studies being done and being approved, certainly those pathways now exist by the FDA of all people. I was telling Subhash, I would have expected places like India and China to be up on this because it's been part of our culture for so long, plant extracts. But interestingly, the people who are incentivizing an herbal pathway for prescription drugs is the US FDA, which is amazing, you know, but maybe we can talk about that a little later. So Subhash, I don't know if this answers your question, but these are the main human studies. There's one or two more, but I can't get to the authors to give me their results. I don't know their results, you know. So the animal data is very strong, but the human data is just like, you know, they make you taste it and they make you all excited, but there's nothing definitive. I think that's kind of one way to say.
Yeah, but again, Roy, even though there's a few human studies that you just mentioned on the diabetes and the endothelial function and all like that, but still the parts of ashwagandha that you use, is it the roots or the stems or the leaves or different species of ashwagandha? And so it's still away from telling you which is the exact ingredient that benefits your diabetes.
No doubt, no doubt. So these are the issues, you know, which of these 40 bioactive ingredients, how well is it extracted? If I go to GNC and buy Himalaya Drug Company's ashwagandha, just for argument's sake, I'm not endorsing them, but can I be guaranteed, you know, the 100, 200 milligram capsules that I pick up today will be the same if it affected my diabetes positively and I thought it was due to this ashwagandha extract? Can I be guaranteed if I go back in October and December, it'll be the exact same? So these are very, very critical issues in herbal medicine extracts. The FDA has tried to address this because they created this pathway about 20 years ago. So it's not brand new. They're almost onto the second. In fact, they have published the second guidance to people who are interested in this kind of thing. So in their second guidance, they have addressed this standardization issue. How can we be sure that a plant extract has reasonable efficacy and no more increase in safety for human health, depending on how it's extracted? So I think they have come out with a very reasonable, sort of almost Buddhist way of doing it, which is the middle path, you know, which is if you claim that your extract has, say, 8 to 10% of the bioactive constituents, you don't have any toxic materials, you don't have any fungal stuff, you don't have any heavy metals that could cause human health problems, then will 10 to 12% work just as well, and will 4 to 6% just as well as your 8 to 10%. I'm just giving an example. And if the slightly higher percentages work the same and don't cause any more side effect profiles, that's a big concern, it's okay. They're happy with that. So unlike, say, metformin, which I go and fill in Rite Aid or CVS, I can be guaranteed, doesn't matter who made the generic formulation, you know, unless there was some big fraud, mostly I can be guaranteed that metformin is 99% going to be the same. Some of those fillers and the way the capsule is made and colored is going to be a little different. But the basic molecule is one. The so-called chemical entity is one. Here we are talking 30, 40, and the extraction process would have concentrated a certain group. So the FDA is saying, okay, if you have patents on the extraction process and you're saying it's within a range, say you say between 8 and 16%, just for argument's sake, you will improve diabetes and sugar control. You will improve endothelial cell index measures. That's good enough as long as the 16% didn't cause double the amount of side effects. That's also okay if it causes side effects, as long as it's not dangerous, because some might be just annoying side effects. So this is the way in which the FDA is trying to bring it in. But as you know, this entire business is controlled outside the drug part of the FDA. It's more under the F part of the FDA, the food. So it's more under herbal, botanical, nutraceutical. So to go to the FDA, you're already talking prescription drug and studies done like prescription drugs. Completely different standard. And of course, the cost will be higher because the expectation of studies, these gold standard studies, they're not cheap, which is why there's huge cost sometimes for the single blockbuster drug comes. All kinds of factors are there, obviously, in the pharma industry. But those are very critical issues when it comes to herbal, products and how we use them. The advantage of some of these products is the standardization may not be the best, but because they are in low concentrations, usually, not in every case, the side effects are quite low also. Not always, as you know, sometimes just depending on where it was cultivated, how it was harvested. Sometimes there could be arsenic or lead, all kinds of things can happen. So, that purification process is critical. That's a very important point though, All right, so if I may move on to the next group of things that affects some of us as seniors, as we age, as some of us retain a very sharp intellect and cognition, yet others get affected just by living longer and longer with cognitive issues. And some of it is just age-related memory, age-related concentration, attention. So this is a study. So the Indian government the central government set up this National Brain Research Center. Some of you in the audience may be aware of this. It's been around for 10 plus years. It's in Gurgaon. And, basically it's a complex where they are going after things that could affect India or the South Asian population in a big way. And as the Indian population lives longer and longer, just from the sheer volume, the government has predicted this will be a huge drain if it can impact places like rich countries like Japan and the US and Scandinavia by taking people out of the labor pool and having the government or the taxpayer to support older people. How do we make sure we're addressing this scientifically? So it's sort of one of the mission of the National Brain Research Center is to go after these kind of things. So Vijayalakshmi Ravindranath, she's back. Let me just go back. At the Indian Institute of Science. She was at NIMHANS actually. And then, went to sort of help set this thing up, was there for a decade. And this was some research they did with a specific extract of Ashwagandha approved by this temple in Kerala that has been doing Ayurvedic work for something like 400 years. So they have a very specific extraction. And so she approached them and they said, if you want our cooperation, you can do scientifically whatever you need to do, but we extract it in a very specific way. And so they modified it for scientific purposes. This is not a human study. These are done in mice models. And Subhash and I were talking about this. And, it was published 8 years ago. So the study was done a little before that. Dr. Sehgal, she is the first author on this, but the leader of the group is her. And now, she is not in Delhi and she's back in Bangalore. And she was here in Pittsburgh not so long ago talking to the Alzheimer's Disease Center. So at this point, given Subhash's interest, in pathology, I'm going to hand this off to him for the next couple of slides so he can explain. Shall I mute myself for a minute then?
Yeah, all right, Roy. It's very, very, very interesting to me what's going on. And I think still we have a long, long way to go as yet on ashwagandha. But this part of the slide is a pathology slide, so it interests me because I can read it pretty clearly. People who are not familiar with this thing, and I'm sure many people are, let me just explain a little bit what these slides are. This top row and the bottom row, the top rows are from the what they call the brain cortex slide. The CT is a cortex, and HP is the hippocampus section of the brain. There are different parts of the brain. Hippocampus is mostly involved with the memory part of it, and the cortex part is more like cognitive functions of the brain. And so what you're looking at here on the left-hand side of the slide, which says vehicle, is the untreated patient or the animal model that you have, cut section of the brain and stained with some special stains to highlight this little granularity and small deposits you're looking at, all the brown stuff that you're looking at. These are what they call in Alzheimer's disease, the amyloid plaques, or the neurofibrillary tangles, they call them. So these are kind of a thing that is the pathology part of what leads to Alzheimer's, because all the neurofibrillary connections and synaptic connections are all disrupted by these tangles, and they all messes up the whole nerve system, in other words. So all the memory part, all the cognitive functions, and many of the nerve functions are disrupted by this plaques. And same with the hippocampus. You can see these small brown things over here too, which is the pathology part of the Alzheimer's disease. And after treatment with this extract, these things have disappeared, or maybe much reduced over here. And so is on this side too here, even though there are small plaques still left over, but much of the stuff appears to be reduced or improved. And when Roy showed me the slide, it just struck me that, my goodness, this is the Alzheimer's disease they're talking about. And if the extract alone can just simply reduce your thing in the animal model, just imagine how much potential is there for the human aspect of the disease. Alzheimer's disease is a widespread thing. Many drug companies are after it. And if they can simply purify this ingredient and maybe, synthesize it and standardize it and use that for the human experiment, there's a tremendous potential in the market also, and also for the benefit of human beings. So this is what impressed me. So I think there is lots of ways to go as yet. And if this is true, what you're having, Roy, I think if you can explain to us a little bit like how this disease was created in the animal model to begin with, and then how this experiment was done, like how many weeks, how many months of the treatment to read this result here. And same thing here. This is another slide where you have, again, the vehicle is a test model, no treatment, all the brown stuff is the disease. And oh my goodness, 7 days, 14 days, and 21 days. In just mere 3 weeks of time in the animal model, as you can see the reduction in the plaques here.
Yeah.
Impressive. I'm very impressed with this thing, Roy.
And so, it's a very difficult journal to get into, the Proceedings of the National Academy of Sciences of the US. And this was published, 12 years ago, because it has such a different mechanism. Because I think a lot of the drugs that you mentioned that have failed, the big companies, Lilly, Pfizer, Merck, they've all gone after Alzheimer's drugs. They've gone after antibodies to go after plaques and tangles to try and dissolve it. This particular study, is done in the so-called APPS1 mice. You can see it in the heading here, And there are many mice models and rodent models of Alzheimer's. They don't necessarily translate 100% to humans naturally, but they have been used to study what could be not just Alzheimer's disease, but what could be the drugs that might help prevent it, or once it's established, how do you reverse it? Most people have gone after things that create the plaque or create the breakdown of the plaque in the brain. This study is interesting. There was an editorial written in the same issue of the journal where this was published, because I think of the mechanism. People did not think the cholesterol receptor in the liver could have something to do with Alzheimer's. And this particular model of mice, this genetic model of mice, they are bred in such a way that they develop plaques and tangles, as Subhash was saying, very quickly, like within what they call the mice middle age, the mice old age, and then they have a mice old age. Because, the lifespan of these mice are different. And of course, these are, experimentally bred mice just for this. And there's several varieties of, models where this can be demonstrated in fast time. But interestingly, what ashwagandha, this extract seems to do in the mouse liver, it speeds up this LDL cholesterol receptor in the liver. And somehow the extraction of that, these eventually goes through the blood-brain barrier, and it extracts it out of the brain into the, circulation, brings it back to the liver and breaks it down. It is so different from anything else that has been mechanistically thought of. And why this hasn't gone on to full-fledged studies, it's unclear to me. Maybe, I know the Indian government, the National Brain Research Center, has taken out patents on it. I think they're worldwide patents. They've assigned the patents to the government, so that, there is, even if the government assigns it to someone else, at least they're involved, and maybe there's some way in which it can be made available to the poorer person. I don't know if that's the intent. But in spite of that, it's shocking to me why big studies haven't been done, either in diabetes or in Alzheimer's or in mild cognitive impairment. If the argument is in established Alzheimer's, you really can't reverse it. You have to start earlier. But, now we have even defined earlier as mild cognitive impairment with a set of criteria. It hasn't been done. You will see the story repeat itself in my presentation, that human studies have been inadequately or not done, because probably, there's some natural skepticism in many parts of the world for natural products, chemistry, and use. And the other is the interest level of individual research groups to go after it. And maybe the lack of incentives if there's no intellectual property protection. So there may be a combination of factors. But the same book, just to continue the story, you can see just that one group of 40 to 50 bioactives, this particular group that I had mentioned in one of the medicinal structures, was about 40 plus withanolides. They seem to go after so many different areas, and we have been just touching diabetes. We're talking a little bit about neuroprotection and Alzheimer's, and there's a whole bunch of others. There's definitely for tropical diseases, especially bacterial. There's now viral in the immunomodulatory part, cardiovascular, and certainly cancer, right, in both areas. So there's tremendous interest. And so what about cancer? In that book, I told you a whole section. This is the work of other people. I don't have any claims to any expertise in this. But animal data in cancer is sort of like the diabetes data. There's a huge plethora of work for decades. Different laboratories, different countries, including right here in Pittsburgh with Dr. Shivendra Singh's group at Hillman. I told you about the Japan book, so there's a lot of that. And so clearly there's a lot. And just to give you one example here from Pittsburgh, from Dr. Singh's lab, certainly he could speak to this way more than I could, but if you just look at this figure, a particular type of animal model, these cells, they grow fast, especially in what's called a particular nude mouse model. And if you look at the weight of the tumor, this is the mouse not given ashwagandha, and this is the mouse given 4 milligrams per kilogram of Withafrin A. So you can see the weight just in terms, it's almost like cut in half. This is somewhat harder to see because we have created like little targets around it, so that, because otherwise it would have been hard to point out to a 100-person audience, in Zoom. So I just had, my assistant Joan make things, but you can see the control at 12 weeks, not fed ashwagandha in the diet. And here it is, right? And so you can see when you give administration of this particular synthetic part of ashwagandha, Withafrin A, the first chemically synthesized molecule out of the ashwagandha plant, on this particular model, what happens is, you can just see like what the target looks like in the nude mouse model, which has had the grown tumor transplanted under its skin, and it just grows rapidly. And here it has been fed ashwagandha and it grows way slower. So these are just examples and just a flavoring for you as an audience to what's going on if you're not already in this field. Similar data out of Japan, ashwagandha, this time, same nude mouse model, and they grow these tumors and then transplant them under the skin. And if you're able to see it here, certainly in these circles, they're very obvious. And if we look at the days 8, 11, 15, 18, 21, so this is control, not fed ashwagandha, just normal mouse food, fed a rich ashwagandha withanolide. It's very rich in withanones, especially. You can see the difference in the size of this transplanted under the skin tumor, right? So what about human trials? The same question that, Subhash had asked a little earlier with diabetes, very little work. Dr. Biswal, Malaysian trial, 100 patients with various stages of breast cancer, usually starting, I think, at stage 2 and above, undergoing chemotherapy. And it's not a practice that I have, but I certainly, I've had a cousin who's passed away from this. And she always told me that after the chemo, in Chennai and Bangalore, she always had like a chemo fog. She couldn't think straight, and even in the work she was doing, it was harder, and the co-workers would notice. Of course, her family would notice. And even when I talked to her, I would notice, but I knew better not to make a big deal of it. But, and so this chemo fog can be very troublesome, pain, fatigue, all this can be troublesome. It wasn't a randomized study, other than he just gave it naturally to people 3 times a day, this root powder, a particular kind that he had, and added it to chemotherapy I think it was called the Eastern Oncology Group in Malaysia, for those who just had plain chemotherapy and then chemotherapy for ashwagandha. It's a published paper, and I can certainly send that. It's in the public domain. If you ever Googled it, you can find it, easily. But if you just look at fatigue, pain, emotional and general quality of life, significantly better in this naturalistic observational study. What other cancers? So, I was looking like 8-10 years ago, what's the right dose of the extract we were going to use in bipolar illness? It's a study we did first, and schizophrenia was the bigger study we did. And how far can I go in humans with the extracts we had? So I reached out to different people where people were doing some ashwagandha studies. And Manish Agarwal's name came up because I was searching in clinical trials registry. And his name came up and he told me he had spent some time here in Pittsburgh and had just gone back to Mumbai. In those days, I think he was still at the Tata Memorial Hospital. Now he's at Hinduja. And he had, for late stage bone cancer, osteosarcoma. He's an orthopedic oncologist. You know, he said, a lot of people don't want any more chemo. They said, no, I just want to die. So he had heard just from anecdotal use from Ayurveds and others, you know, what's wrong in giving people strong immunomodulatory anti-inflammatory compounds like things that have come out of, say, turmeric or ashwagandha. And so, you know, he found like a certain oral preparation of ashwagandha. I think it's a Mumbai-based company. And he formulated, I think it may be more than ashwagandha. It may even have, if I remember correctly from many years ago, it may have even curcumin extract in it. I'm not 100% sure. And he just wanted 24 patients who had given up on going back to chemo and had kind of given up on life. And as you know, this particular bone cancer can be pretty aggressive, and it goes from like teenagers all the way to older people. So, and it has never been updated. I've tried to reach him about 3-4 years ago. I couldn't, you know, in the early days, I could get him easily when he was at Tata Memorial, but now I haven't been able to find him. And he told me he had spent some time here. I don't know if it was with Dr. Shivendra Singh, but he had spent some time in Pittsburgh and had just gone back. So we were just, you know, remissing why we hadn't met when he was in Pittsburgh. But anyway, to go on to another human clinical trial, just begun, not yet recruiting fully. In Sacramento, Dr. Stacy DeAndre is going to study a particular extract of ashwagandha, but mostly for this chemo fog I was talking about, where it affects your fatigue, your memory, your concentration, maybe you get depressed. And since ashwagandha has these properties to go after all that, and the Malaysian study was encouraging, she's doing a placebo-controlled study. It has just begun, but of course, with the pandemic, you know, a lot of research has just come to a grinding halt. And so has this, I imagine. It certainly has here in Pittsburgh. And so, you know, we'll see what the results look like. But again, for the amount of animal data in different kinds of cancers, very little modern kind of data that we've come to expect. Now, maybe in the Ayurvedic world, there are different kinds of studies that maybe it's not in the published literature, not easy to come by. I'm unaware of that. Not that they may not exist, but I'm unaware of that. The next thing, Subhash, should I just keep going or did you have any questions?
I can maybe ask a question here if you don't mind.
Sure.
Despite the limitation on human trials, as you mentioned that very, very little studies exist on the human trials, but nonetheless seems to be encouraging to see that these trials are gradually increasing now, I think. I think people have taken more interest now. And why do you think, in your mind, that there has been such a slow pace? Because you have shown quite a bit of data already on the animal models that the big pharma would like to jump into it and spend money in finding out the exact ingredients and the dosage and maybe synthesize that molecule and be more specific and go after that. I mean, why is it not happening like that?
I think multifactorial, you know, when Dr. Bhushan Patwardhan, who I mentioned, you know, has that patent for that adjuvant vaccine with diphtheria, whooping cough, and tetanus. And he has done a lot of work, and he's the editor of this Journal of Ayurvedic Medicine. And, you know, he always wondered, and I sort of agree and concur with some of his views. I think multifactorial. One is, how do you say what does what, you know, of these 40 bio things. Right. In modern scientific training, we are not used to having multiple things and not having the mechanism behind it quite clearly understood. And in the scientific, especially in the medical community, that is very suspect not to have that level of specificity and standardization because we've come to expect that. So that's one. The intellectual property protection for these. So for instance, Chantix is a Pfizer patent for smoking cessation. It has a certain bunch of neuropsychiatric side effects, but really in Central and Eastern Europe, they were using the plant from which cytosine was extracted, and they were selling it over the counter in their own little pharmacies way before the Pfizer scientists here in the States pulled out varenicline, the single chemical molecule. That was almost 10 to 15 years later, and did it. It's the same story for aspirin. People in Western Europe 200 years ago were chewing the willow bark because they knew sometimes the swelling in the feet would disappear, but they didn't know what caused the swelling. For some, it was congestive cardiac failure. For some, it might have kidney failure. But they just chewed the bark, and for some people it worked. It didn't kill them. So eventually, Bayer scientists extracted aspirin out of this willow bark. And of course, they got, a patent. And look, 100 years later, where aspirin is, 150 years later. Digoxin, another plant extract. We can go on, lots of tropical plants. Initially, they're just used in the communities till these synthetic molecules are found. But Bhushan Patwardhan would often argue with me, and I wouldn't disagree. Actually, I have the same sense that when you make it super specific, you will have a severe side effect sometimes as the trade-off, granted. But mechanistically, it's something that is in our comfort zone, in our training in science and understanding in medicine. And so we have come to accept that position. So the position of these multiple bioactive constituents, why is it working? Is it just a fluke? This is what leads to that. And if there's no intellectual property protection, because of the millions having to be spent in the preclinical phases to develop it into a prescription drug, it just dies. So many factors, I think. But interestingly, the FDA has actually created an herbal botanical pathway. And as I think I may have mentioned this to you, Subhash, only 2 drugs have been approved in the herbal botanical pathway in the last 10 to 15 years, because a lot of the companies don't know how to do these traditional clinical trials. You're held to the same standard if you want to make it a prescription drug, just like any other allopathic single molecule. So these are some of the issues, as to why such a solid amount of animal data, very suggestive, and you keep scratching your head, why isn't someone going after this gold mine?
We have one hand being raised by Dr. Rajnikant Popat. Okay. And then after his question, Roy, then we can continue with your talk, and before we take the other questions after that.
Okay.
Okay, so Rajnikant?
Yeah, Dr. Chengappa, thank you very much. It's a very interesting topic in light of the fact that commercial entities are running faster than the amount of research that is being done. And from your talk, I am understanding that there just hasn't been enough of effort to fractionate and standardize each of the 40 withanolides that you talked about. Because we don't know which one has pro and which one has anti-effect, and the sum total may be potentiated or decreased depending upon the component that is predominant in that particular extract. So my question to you is, are there any studies where they have individually looked at every fractionated component to really see which one has effect, potentiating effect, or anti-effect? And the study that, the other study that Subhash presented about the amyloid deposits.
Yeah.
I just couldn't get the correlation between withanolides and this mode of action in reducing the amyloid plaques in the brain.
Yeah.
And so, if you comment on both these components, I will really appreciate that.
Yeah, I think it's like a very crucial and critical question, really. It's sort of what Subhash had got into and what you've done very specifically, sort of what we've struggled with actually for 10 years. So Withafrin A, that original synthetic compound, because it was in the 60s, my suspicion, though I've not talked to industrial sort of, grade people who can manufacture, I suspect Withafrin A can be manufactured beyond just laboratory manufacturing, all the way to commercial manufacturing. Withafrin A is one of the most studied compounds because it was, I think, started early, very potent anti-inflammatory, immunomodulatory effects. My hunch is, Dr. Shivendra Singh's lab here does much of its work with that, whereas others. So with the withanones, I think the ability to synthesize creates very standard single molecules or 2-3 molecules. Not that it hasn't occurred in animal work or in vitro data. I think the trick is how much can be used in humans with Withafrin A. If on the one hand, it turns out to be like a chemotherapeutic drug, very toxic to the bone marrow. So these kinds of studies would have to be done at a very basic level. So genotoxicity studies to make sure that DNA is not altered, have been done with single extracts, single chemicals even from the plant, and from like bioactive crude extracts, fractionated. Definitely they've been done. Human studies, phase 1 studies in the classic way, have run into issues like which of these 40 bioactives should we study for which, and can we study the pharmacology of it to say, okay, it can get into the heart, into the liver, into the brain. So that kind of very basic pharmacology, human pharmacology, is missing. So a lot of it is, I think, driven to some extent by how much intellectual property someone has to be incentivized to invest in somewhat of a high-risk proposition. The animal data are very clear, points in the right directions. Early human data used in a naturalistic way, but from somewhat unstandardized preparations, also points in the right way. If we become very specific and get, say, just Withafrin A and say just for cancer, will it end up being like any other chemotherapeutic drug, suppressing the bone marrow, decreasing the white blood cells, making you more prone to infections? We don't know the answer to that. But that's the kind of way future studies will have to go. If, and it's happened with other tropical plants, we already have individual chemicals from it with pluses and minuses, like you were saying. Some will have pro effects, some will have antagonistic effects, and some will have toxic effects even. So this is the holy grail of tropical plant chemistry, all the way to a prescription pad eventually. So we are still, I think, not there, but the data I will show you, for instance, and this is not to endorse just one particular extract. We have used this particular extract. Natrion is this company in New Jersey. So they have done studies themselves. This is done in an Ayurvedic hospital with what they call a standardized extract, not a single chemical entity, not one molecule, but a way in which you process the extract, and they have a bunch of US patents for the processing. So in this Ayurvedic hospital, anyone who expressed stress, I guess mostly in Bengali, however stress was expressed, was, and for whatever reason, could have been work stress, could have been life stress, could have been fighting with the husband stress, whatever, however it was defined, they went to the Ayurvedic practitioners, a totally Ayurvedic hospital, and they were given the sensorial extract, 125 milligrams per day, 250 and 500. About 30 and 30 people here, 19 people for this lower level, and placebo about 15 people. Blinded study, Dr. Vishwas Audi and his team did not know who was getting what. And it was done for 60 days, 2 months. And this is a scale that we use, the Hamilton Anxiety Scale. And just to orient the audience, if you come down, you're doing better. At 20, 25, you have moderate to high level anxiety or stress, according to the scale. It's a scale that has been used in psychiatry. You know, Max Hamilton was a psychiatrist in a big hospital in Britain, and he came up with HAM-A, Hamilton Anxiety Scale, and HAM-D. Because psychiatry, we don't have any objective markers, we use these scales. And the scales have been validated. So clearly you can see, I mean, quite a robust decrease in the stress as in this modified Hamilton Anxiety Score. They modified it for the Indian situation because not all English language questions translate fully to every Indian language. So it was a reasonable modification, but it was a double-blind study and showed this. So this is a standardized extract, but standardized with the caveats I mentioned. They can say it is, they have a minimum of so and so percentage of ethanolides and very small percentage of Withafrin A. I will show you in my studies why that might be important. But this is just a flavor. Again, there's about half a dozen stress studies done by different groups, not the same group, same extract. The Canadian study, I don't know if I have it here. Maybe not. But anyway, if I come to anxiety, I might show you a couple of different studies that use different extracts and sort of ended up with the same result. Cognitive improvement in bipolar. I've just put two of our slides. So this, of course, is something we did and published long back. It was funded by a foundation that, you know, funds people who want to do studies which nobody else will fund in schizophrenia and bipolar, a little bit in depression. So we were funded by what was called NARSAD. It has a new name now, the Brain Behavior Foundation. And they fund studies with small grant, $100,000. And we did 60 patients roughly. And eventually we had this many people on Ashwagandha and this many on placebo. So what is digit span? What does it mean? So for those of you who are used to cognitive testing, you will know what this is. So if I tell the audience, repeat after me, 0, 1, 3, 5, 7, 9. I've given you these 6 or 7 digits, and I will say just repeat after me. Most of you have already used your association memory that I've gone by every alternate number, and you have already figured this out, and you will just repeat it in next to no time, right? So this is the forward digit span. So it reflects how attentive you are to what I'm saying. Maybe you're not playing around with your iPhone, I'm just kidding, you know, or not taking a sip of coffee, but you're paying attention, and it's your attention and concentration that we're measuring. Now, if I tell you, now try and tell me that backward. And if I mix it up, I don't give it in this order of 0, 1, 3. Instead, I tell you 1, 5, 3, 9, 7. I sort of mix it up a little bit. And then I ask you to repeat it. Now you have to say, okay, I put it into my memory. It's there in storage, temporary storage. I'm now asking for you to repeat it backwards. So this is your working memory. This is how you remember your 7 digits, if you leave the area code out, the phone number. So this, you and I and children by the age of 10, most normal children, will have about 7 plus minus 2 for forward and maybe 1 or 2 less for backward digit span. So it is a function of our attention, concentration, and working memory. And we have to have reasonable hearing so that you heard my instruction. So to orient you to that background and what this means, on average at bipolar patients who were stable, attending outpatients, coming only 3-4 times a year to 2 of the clinics at Western Psych in Oakland, had about 5 backward. And 2 months later, at 8 weeks, the placebo went up by 0.2. The Ashwagandha group, remember this is a double-blind study, this is an objective test. The objective test doesn't know who's getting Ashwagandha, who's getting placebo. Almost goes up by 1 number. Now, for me as a clinician, I don't know what this means, but I asked some neuropsychologists. They said it is amazing to go up by one whole digit almost in 2 months. Almost, you can't even do that with brain exercises. And so, it was a modest effect. It wasn't like earth-shattering. We put it into the literature. We had hoped for bigger effects, but we got 3 effects in people. We didn't touch any of their other medicines because they were stable people. They were just complaining of cognitive issues, typically people in their late 40s, early 50s, with about 20 years of illness, and stabilized on their whatever the mood stabilizing or psychotropic or antidepressant medicine. We didn't touch that. We just added to people who had complaints this, and we did before and after tests. Many of the tests did not come out positive, but this digit span was very striking. And so this was in the literature. What I was far more interested in was its anti-inflammatory properties in schizophrenia. And I was looking for someone to fund that. And so I went to the Stanley Medical Research Institute, which is Washington, D.C. based. They have offices in D.C., Maryland, Virginia, and they have funded studies, one branch of the Stanley Tools family. And, they have a son who has bipolar illness. The parents have passed on now, but, they have given $250 million worth of funding for 25 years for schizophrenia and bipolar research. And they are the biggest private donors to the Broad Institute at Harvard and MIT. They gave $1 billion when Mr. Stanley died. He gave $1 billion for research to that institute. But anyway, so I was the recipient of a small grant in schizophrenia from that group. And to orient you to this slide, so schizophrenia, so bipolar illness is a mood disorder. You have up, you're very happy, beyond just very happy, you're almost manically happy, doing crazy things, spending too much money, wasting away your family fortune, taking your college kids' money and giving it away to people, talking too much, not sleeping, not eating, trying to tell Mayor Peduto how to run the city, and so on. So this is mania. Depression is depression. We all understand depression. So that is bipolar illness. The classic example of that is often in Bollywood and Hollywood. The arts and entertainment industry has a lot of people with not full-fledged bipolar, but some traits, because it also comes with creativity, with the ability to just sleep a short amount of time and do everything. So unlike many psychiatric illnesses, bipolar illness, when you get well, you can run a country, a company, and everything, right? And you can have your life back and do even more than most human beings. Schizophrenia is a very different illness. It hits you somewhat young, college, high school, destroys for many people their life trajectory. They're hearing voices where there are no voices. They're becoming paranoid about things where there's no need to be paranoid. They're talking in a way nobody can understand and out of touch with reality completely. Some extreme manic patients also have this in bipolar illness, but in schizophrenia, this is the defining feature. But when they get better with the antipsychotic drugs, all that improves. But what they're left with is what we call negative symptoms. And that looks like depression to the common person or to the family. They are simply not doing anything. They're sitting at home, not willing to get up, even if they're college educated, even if they're working, not willing to get out of bed and brush their teeth. So you're saying, what's going on here? Why don't you just pull up your bootstraps and do something? And it looks like depression, but it isn't, because if you actually talk to them, they're able to talk back to you and they don't come across depressed in any other way. And they will tell you, no, I'm not depressed. It's just this so-called negative symptoms. Just like the positive symptoms are so stark and it is something positive that comes out of a brain, but it's all unreality based. Negative symptoms are as if you've lost your humanness. You don't speak, you're not motivated, you have no energy. And so this is a syndrome that we often have to tell families, this is not just laziness. This is just part of the syndrome, and for that, none of these antipsychotic drugs work well. In fact, they're useless half the time, 3/4 of the time. So we need something to improve it. Some of the therapies are really good for negative symptoms, or for depression and anxiety or demoralization that might come after hospitalization. And so, of course, we have been trying to get all kinds of medicines. The FDA has incentivized the pharmaceutical industry to come up with something for negative symptoms or for this cognitive impairments also, because in a way, they impact your functioning in life. There, we got this somewhat surprising result with ashwagandha. So to orient you, this is the ashwagandha line in yellow, right? And the purple line is the people who got placebo. Roughly 30 patients in each group, about 33. In each group. We studied people in our clinic over about 2.5, 3 years just to recruit these 60-odd patients. And so if you look at the placebo and negative symptoms, they improve a little bit, then they flatten. This group, by visit 3, which is 2 weeks later, has sort of begun to improve, but not clinically or statistically. By the time they get to 1 month, significant improvement. It keeps on going. We stopped the study at 12 weeks. I don't know if this could have kept coming down further. We don't know. This was a double-blind, placebo-controlled study. We had, because I'm doing it through PIT. PIT insists, even if it's a herbal, I have to do it as if a pharma company is doing it. I have to go to the FDA, get approval, all this kind of thing. And then how about general symptoms, not psychotic symptoms? Of course, that is almost like negative symptoms, depression, anxiety, motivation, similar result. Nothing happens till about 1 month. At 1 month, the effect starts. And this is total. This is sort of like a cheating slide because this adds up this and that. Naturally, it's going to be the same. Positive symptoms, we did not expect it to be an antipsychotic. It's not, in our opinion. There was a trend, but it wasn't clinically improving. So again, we used, as the question was asked about standardized extracts, we used this particular extract, which is standardized. I'll speak to this issue a little more, a little later. Another study, Mumbai study, right? What about for sleep? Ashwagandha has certain constituents which are seen as great for restorative sleep. It's not like benzodiazepine. You take it, if you haven't taken it ever, you will fall asleep. You'll almost be in a coma because you're not used to it, unless you're someone who abuses medicines, right? So here, a different kind of extract, but also double-blind, randomized, and placebo-controlled. And to orient you to the graph, everyone in the placebo arm in blue, or the ashwagandha arm, is falling asleep in a sleep lab about 45 minutes into getting into bed. In 5 weeks, this group has improved slightly because they're all being taught sleep hygiene also. But the ashwagandha group has separated big time. It has dropped from 45 minutes to about under 30 minutes. You might think, what is the big deal in 15 minutes? Actually, in sleep research, it's a huge deal, and then it has dropped to almost like under 25 minutes. So your sleep onset, the latency to falling asleep and simply being restless has dropped a lot. But what does that mean? Does it mean much if you're not going to be more efficient in your sleep and you're just getting up in the middle of the night instead? That measure is called sleep efficiency. And here the orientation on the slide for the audience is the opposite. Everyone starts around 70-odd%. In 5 weeks, the ashwagandha group has got to 80-odd%. And here, it's almost at 85% at 10 weeks. The placebo group that has been taught some sleep hygiene measures also improves, but these are statistically and clinically big differences. Here's a scale that is anymore used all over the world. It was invented in our department. It's called the Pittsburgh Sleep Quality Index. It was invented by our former chair, Dr. Kupfer and Dr. Baisi, and it is freely used not just in academic research, in all sorts of studies all over the world, translated into some 50 languages and so on. Same thing, in the double-blind study, you can see this huge improvement when it comes to ashwagandha at 5 and 10 weeks. Orientation for the audience, going down is better. So this, interestingly, in our bipolar and schizophrenia studies, spontaneously, even though we didn't measure this, we would write down statements that patient made in the 2-month and in the 3-month studies. A lot of them would say by the 2nd, 3rd week, 4th week, typically, almost the same time frame, that their sleep feels better, more restored. Some would simply say, I'm sleeping like a baby. And when we broke the blind, definitely those statements were far more likely in the ashwagandha arm than in the placebo arm. So in psychiatry and in related fields, we have definitely had more randomized double-blind studies. Not with single molecules, but with standardized ashwagandha extracts. How about for another common thing that a lot of us have as seniors? Aches and pains in knee and shoulder, and elbow pain, wrist pain, right? Various reasons. Sometimes arthritis, sometimes something else. Study done with the standardized extract back in Hyderabad, the same person who had done those diabetes studies, it's her group really, it's this group, Usha Rani. She's like the head of the department. She does some good work with different plant extracts, including ashwagandha, and even though the legend here says 250, 125 and placebo, it's twice a day. So this group is actually getting 500 per day, A, and that's this group. B is 250 milligrams per day. It's just the 125 milligrams strength, but given twice a day. And placebo is getting it also twice a day, and that's C. Here, to orient you, it's actual knee swelling of how much on a scale of 0 to 10, the patients who are blinded, whether they're getting placebo, low dose ashwagandha, high dose, are voting with their feet and saying how much the swelling went down in 3 months. Their rescue medicines are allowed. If they were taking Tylenol, paracetamol, whatever, they're allowed to continue all that. You can see the swelling reduction. I just took out 2 slides from this paper. There's lots of data in this paper. It's a published study. Now, this is the journal that Bhushan Patwardhan edits, by the way. It's not an easy journal. They're tough reviewers. So it's a good journal, very well sort of written and driven. And then here's another one. Also by blinded, until the blind is broken, nobody knows what they're getting. This high dose ashwagandha group has the almost like 35% improvement in pain scores compared to the 250 group and compared to placebo. So these are human studies, standardized extract, but not single. As Subhash said, I just put these in so that when you go online and look at, ashwagandha containing products, the ones we have used have contained sensoril. These are things that we tend to see. And when patients ask us, we have to be careful in what we say because we don't want to be seen as selling the sensoril product. We just say we have done one study, and one study is not enough, more studies, someone else should replicate it. We say the things we should say, based on our training. We can't simply go and say, do this and do that, But how about other ashwagandha products? There's plenty on the market. I do not know the characterization of these products simply because in addition to my day job, in an administrative capacity or in a research capacity, I haven't had time to study all these other products or to use them in our studies. There are even liquids for those who might not have like swallowing problems or teeth problems or children, and so, but I have no idea which extracts and how well they are done. I have no idea about that, but they're there, So back to something that you brought up. These are critical things in standardization in herbal medicine. we can't emphasize this. It's very, very important. Our particular, brand of ashwagandha we have used in our studies, they've been concentrated to at least 8%. So if I'm saying I'm using 100 milligrams of this extract, what I'm really saying is only 8 milligrams of that 100 milligrams has what we think is particularly important. Maybe for brain diseases, psychiatric diseases, maybe we can't have very high rates, concentrations of Withania, because they might cause more toxicity and more side effects. They may be very important to reverse this for certain kinds of cancers. So that's the kind of study that's not been. This particular extract has oligosaccharides, which is really sugar. As you know, saccharides are just sugar molecules. And we feel, and including the people who supply us this, and I've talked to the R&D scientist, Dr. Ghoshal, he feels these are carrier molecules that will take it into the brain. And that's why it's important to have a high concentration. This we don't want, because this would add to the toxicity and side effect profile. So this is standardized in that kind of way for our studies. Other issues, in addition to where it's grown and the pollution or what's there in the ground, there's a lot of chemotype. Just like we in humans, we have genotype specificity. Plants have a lot of specificity in cultivated versus wild plants. Batch-to-batch reliability, which we have already addressed in questions. How much is absorbed? How long does it stay? Standard things that we tend to ask in medicine, not well known outside the animal data. What is toxic and what is really useful? What might be the mechanism of action? What drug interactions might it have? And how good are the control trials? My thoughts of 10 years of doing this, what is the right way to study them? We can get into this Q&A part. What are the modern allopathic medicines? How well do they combine with these extracts, which have so many, what is doing what kind of raising our eyebrows, So that certainly we have a problem with. How do we standardize so that when we say we are buying this batch, it has already done something for my joints, that each time it's going to keep on doing it like it does for my blood pressure medicine or for my cholesterol medicine? Does intellectual property help or hamper the development and innovation in these? And, so we also have to be careful. Suddenly there would be the 6 o'clock news, right? Oh my God, these Ayurvedic products, they came from this particular manufacturer in India and they're downright dangerous. They can, destroy your liver. And people will get very angry very quickly. That will just put a complete break in the research that goes through with these. But in my opinion, all told, Ashwagandha, I think, is one of the gifts from India to the world and will continue to provide, I think, all sorts of gifts that will never stop giving. That is my sort of hope to some extent, and optimism. But I truly believe, just based on what I'm seeing, it will provide a lot of benefits down the road, maybe 10 years from now, maybe when some of us are dead and gone. But I see a lot of potential for this plant and what it might do for human beings, period. So that may be my last slide, Okay. So I'll give this back to, Subhash, to you or to Chetan.
Yeah, fine. Roy, thanks very, very much. This was an excellent presentation that we had. Now, before I open up for a few questions that we have, I see a few hands raised. My one question that I have before I pass it on would be, are there any side effects to this, generally a safe thing, I would say, that has been used widely by many people for many things all the time? Are there any side effects to this ashwagandha?
From the literature, the kinds of side effects are the ones based on the potential mechanisms of those bioactive things. So say you're on aspirin, Plavix, because you've had a heart procedure, this, that. Ashwagandha's extracts, both the withanolides, withanosides, and withaphyllinate, have potent properties when it comes to inhibition of platelets, which could be a good thing, but too much of it could lead to bleeding tendencies. So there's that one particular drug-drug interaction. Because it acts on the brain, if you combine it with psychotropic drugs, as we did in the bipolar and schizophrenia studies, it could be an add-on side effect, too much sedation. We have certainly seen a little more sedation when people start, but luckily for most people, it's not permanent with ashwagandha. When you titrate and go up on the dose, you might see it for a day or two. I would call them annoying side effects rather than life-threatening. There are potential drug interactions which haven't been studied well in humans. In animal data, there are arguments that it may not interact with the vast majority of prescribed drugs, but there could be different kinds of drug interactions. It has potential benefits for thyroid. So if someone is already on thyroid medication, can it push them into a thyrotoxic stage? Hyperthyroid, not high. Hypo is the predominant situation with the thyroid, the underfunctioning thyroid. And overfunctioning thyroid, luckily, is much less common and rare. But there's one report in the literature of a Dutch lady going to Sri Lanka, picking up this very concentrated ashwagandha extract, taking it for 6 weeks and becoming thyrotoxic. One report in the literature. So we have always in our studies monitored thyroid profiles, and what we have found is those who are like borderline almost become normal. We have not seen people go to what happened to that Dutch lady, but theoretically that is indeed possible. There is an argument in the literature, can it worsen autoimmune diseases and cancers, even though it's being studied fully, as I showed you, for different cancers, because of its immunomodulating properties. This is a hard question to answer, because it's not so straightforward with ashwagandha. It's just black or white. It does many different things to inflammation and immunity. So it's very hard to answer a straightforward question like that. It can interfere, as I think when you and I were talking, Subhash, recently, there is an article put out, I think in JAMA, many years ago, from a laboratory in Texas. I think Das Gupta was the first author, of how it interferes with a certain kind of digoxin acid. And that's very important because, of course, there are replacements to digoxin today. It's less and less used, for cardiac, but still it's used quite a bit around the world. It can interfere and artificially make the level look low or look high. And that's very dangerous with a drug like digoxin because it has a very narrow index for toxicity. It's the so-called narrow therapeutic index drug. So in these situations, one has to be a little careful. What we have found common side effects, if you take it on an empty stomach, it irritates the stomach because it has some of these aspirin and Advil type anti-inflammatory properties. So on an empty stomach, we definitely find for those who have acid reflux, ulcer disease in the stomach and the upper intestine, it could worsen it. You have to be a bit careful, and we advise people not to take it on an empty stomach. Not necessarily you always have to be taking with a huge meal, but you shouldn't take it on an empty stomach. It's better to take it with water, juice, a little bit of food, and after a meal is fine also, but try and avoid it on an empty stomach.
Well, thank you very much on that. We had a few hands raised, and I want to also let you know, Roy, that you know very well Dr. Balwant Dixit,
yes,
the chairman of the pharmaceutical department up at the U of Pittsburgh.
Yeah.
He had great interest in the subject that you had. I believe he's in the audience also. If you are there, Dr. Dixit, you can ask a question if you want. Dr. Dixit, are you there?
I think Mr. Kapadia also had raised his hand.
Yeah, I saw that. I'm coming to that. Yeah. Now, Dr. Dixit had, in fact, Dr. Dixit. Yeah, we are here. Yeah, Dr. Dixit, I know you had a big comment on what you had on this topic. So be brief on the thing, Balwant.
I first want to congratulate Roy on an excellent presentation.
Thank you, Dr. Dixon. Thank you.
My 50 years of involvement in pharmacology has made me a skeptical person. I spent 6 years in a medical college doing research on Ayurvedic medications.
Yeah.
We tested about 40 different preparations in animals, humans, and we had a big group, including Ayurvedic practitioners, MD people, pharmacologists, pathologists, all in a group. We had unlimited budget, but after working for 6 years, we found that none of the preparations we tested had any significant beneficial clinical effect. We did animal experiments, human experiments. So I have become a skeptic about plant-based crude preparations to be used in humans.
Yeah.
So ashwagandha is no exception. If you see some of the structures of the chemicals, they are cholesterol-based steroid molecules. So glycoside, for example, would interfere with digoxin, because digoxin is a... so your anti-inflammatory effect, which you have observed, may be because of the steroid preparations, which are the basis of methanolides.
Yeah.
It might be converted to slight amount of cortisol or hydrocortisone type of compound. Right now, we have very potent anti-inflammatory steroids available, like dexamethasone. So my one question to your research was, you tried somebody else, or you have tried Motrin and all those drugs. Did you try to use one of the potent steroid anti-inflammatory as a control?
To my knowledge, I think
people use a lot. Okay, if they want it and have it, we just take the glasses. Okay.
Could I?
Yeah, please, Roy, go ahead.
Yeah, so steroids have been used in very old studies out of India for animal-based arthritic preparations, and definitely they improved it because, I mean, it's a clear-cut mechanistic way of going about it. And in those ashwagandha preparations, we are talking of 1960s and 70s. I had to get those articles. I couldn't get it on PubMed. I couldn't get it from PIT. I got it directly from the library in Delhi. You know, the librarian sort of sent it to me from the 1960s. It's very interesting. Radiographic pictures of using steroids and that particular ashwagandha extract. And it's quite impressive, really, because I think the difference from steroids and ashwagandha preparations are that in addition to its, chemical lookalike, like a steroidal lactone, it's a, carbon-28 kind of backbone to a lot of these withanolides. And so that's certainly maybe why we have interference with one or two of the digoxin assays and why it might have anti-inflammatory. It has very potent COX-1 and COX-2 inhibition too. So there are those properties of at least maybe the withaferin A and a few of the withanolides. So I think it's able to do it because it's not a single chemical entity from multiple targets. But because of that, I think it's harder to always replicate, and that's why the skepticism comes in with crude plant extracts. But I think with modern lab, studies going on, there is a better way of defining which sets of these will eventually become synthetic molecules that are worth commercially exploiting versus not. And so, I think the new studies, I don't think in Hyderabad they used steroids as a control, but I've seen those animal data from the 60s that used, some predecessor to dexamethasone. Might have been prednisone or whatever those early steroids might have been in the 60s.
Thank you.
Very well. It's almost like becoming a medical seminar over here.
Second question.
Balwant, please be brief now because I think we are running a little bit short of time and we have a couple of more questions to entertain still. So please be brief. Go ahead.
Well, I will not ask any more questions.
Okay, if you don't have any more, then I appreciate that. Balwant, thanks very much for your comments. We have a few hands have been raised over here. I saw Meena Mehta's hand being raised earlier before Shailesh. Shailesh, I come out to you after Meena Mehta's. Meena, you have a hand raised for a while. I saw that. Please go ahead.
Okay.
Okay.
My question is that you did comment about Alzheimer's, but there is also some cognition loss with Parkinson's. Have they studied anything on that? And you also mentioned about solid tumors, but did they study anything at all about blood cancers?
Blood samples of cancer?
So blood cancers like leukemia, lymphoma.
Yeah. So the first part, in terms of Parkinson's, there definitely is a lot of animal work, a little bit of work related to Parkinson's at NIMHANS, at the Ayurvedic unit, things that would be harder to explain, I think, in an allopathic sense. They have a different way of thinking about it in Ayurveda. So animal models, because of dopamine modulation as opposed to pure dopamine antagonism, it almost, is pro-dopaminergic. So when we were using it in schizophrenia, one of our big worries was whether it would undo the antipsychotic effect and actually make it worse. So that is, been a little bit of a concern. But on the other hand, that very property can be useful in Parkinson's disease, and the cognitive effects that come with cognition. So there's been a fair amount of neurological interest, not that amount of clinical work. Animal models of Parkinson's, studies out of India and other countries have shown its benefits for different extracts, and even for, like in the Alzheimer's disease model that Subhash showed those pathology slides. What I didn't show, it's a big paper. The back of the paper talks a lot about those animals when they build up plaques, the genetic models, they get very confused. So if you put cheese at the end of a difficult to get to in a maze, the so-called maze, and you put cheese at the end, they can't find their way. The untreated and unmodified, ungenetically modified, and they will just run to the cheese in next to no time. So this is one. So in this maze learning models, Ashwagandha extracts, the one used by Dr. Ravindranath and Dr. Sehgal, very, very clear cut, in those models. So it's quite interesting, to see its effects in other related conditions like Parkinson's disease, vascular dementia, Alzheimer's dementia. In terms of your second question, yes, been a lot of interest in blood cancers, leukemias, lymphomas, mostly again animal models. And it's the kind of question that maybe Dr. Shivendra Singh's group or Dr. Kaul and Wadhwa in Japan would have a much better handle on than me. Again, I've talked to someone at the Stanford Integrative Oncology Clinics, and he told me he hasn't so much used it for blood cancers as much as he's used it for it's like that study I was talking about going to be done in Sacramento. So he's been using it in San Francisco at Stanford. He's using it along with meditation, yoga, and ashwagandha for people who are willing in the middle of the chemotherapy cycles to improve cognitive function, pain, emotional health. And he hasn't even done a study, he's just using it in practice. And he finds it quite helpful clinically. So, yeah.
Okay, great. So we have, Roy, two more people have a question, raise their hands.
Yeah.
So we'll have two more questions and then after that we'll wrap it up.
Okay.
So the next person who has asked a question is Sailesh Kapadia, and after that will be Abhay Vijli, and then we'll wrap up after that. We'll have Balwant Dixit. I know we have Dalip Desai coming up after that. Okay, Sailesh?
I have a quick question, and the question is, how much dosage should one take of this stuff? I mean, do they tell on your bottle 20 milligrams or whatever?
Yeah. So the dosage, Sailesh, is one of the hardest things because each of the extracts, the amount of concentration they have is different. So I've still not figured out the right dosage. And if I've hit the good dosage, because the way the budgets are structured in the studies we do, we end up with fixed doses. So in bipolar illness, for this particular product I used, which was Sensoril, I told myself, and I took it for, like 3 to 6 months before we gave it to the first patients 10 years ago. And I still take it now. I just take 250 milligrams. But, I personally, in my mind, people ask me the question, the patients or families ask me, what is the actual concentration even within that 250 that's really useful? So I tell them the minimum amount they guarantee is 8% withanolides, which is a whole bunch. So for every 100 milligrams, if I have 8, for 250, I have 20 milligrams. For 500, I have 40, and for 1,000, I have 80 milligrams, even though 1,000 sounds like a lot. The other stuff probably is unimportant, at least for sleep, for stress, for anxiety. So the maximum doses we have used in studies with the Sensoril brand is 1,000. I've asked the CEO of the company in their own studies, how high have they gone in human studies? He told me 1,000. In Hyderabad, they did it in volunteers when they were testing how to measure that elasticity of the carotid or of the radial artery. They just had to see how much human beings could tolerate. And they don't know if people may be able to tolerate 2,000 even. But they went all the way to 1,000 in healthy 20 to 30-year-olds with literally nobody complained of a side effect. But these were small studies, 20, 30, 40 normal people. And then in their diabetes studies, they used that as a model for how high to go. They went up to 250 BID 500. Now, other extracts have been used in anxiety studies, that. So that might come up with a different dosage. That's why when I was giving those product images, I was saying, I really don't know if they're as good as, better than, or worse than the product we have studied.
Very well, Roy. This is a great opportunity for us to hear from a person of your caliber, and it doesn't happen every day for us. We're very appreciative. And so since you are here today, we need to satisfy the few questions that have been raised. So I hear the Durlab also raised a hand. I'll come to him in a minute after that. So at this time, Abe Vijli, he is in the audience. So, Abe, you had raised a hand for a while. Yes, go ahead.
Thank you for your time, Dr. Roy.
Hi.
I have two things that attracted attention, and that is I am suffering with hypothyroid, and I also have a high cholesterol. So those are the two things you mentioned. And I think in thyroid, you mentioned that this, I don't know how to pronounce this drug will either lower your TSH or TCH, whatever that they check the blood sample.
Yeah.
Is that, the question is, will it eliminate taking Synthroid ever? Because I've been told that I'll have to take for the rest of my life.
Yeah. So really important question, and while I don't practice endocrinology, we had great interest in it because when I sent in the first, what I had to create like a portfolio because we are academic investigators, the FDA doesn't give us the same rigmarole as they would give a for-profit pharma company. They were very helpful actually. I had an opposite experience so far, at least with the FDA. So in 2010, when we were applying for the bipolar study, they sent me this report out of Holland. Originally it was in Dutch, but because these agencies communicate with each other, they know these things even before the medical community knows. So this lady who was the head of the endocrinology section of the FDA, she reached out to me and she said, are you aware of this Dutch, this lady who went to Sri Lanka, took this hyperthyroid, and she said, this is just in the reported literature. We don't know of the people who never report. So I said, no, I'm unaware. And She said, the reason I'm calling you is for your study, you are going to have a few people taking Synthroid. I said, of course, these are people with bipolar illness. They have a tendency to hypothyroidism. Many of them will be on Synthroid, of course. We know that. So she said, what we are recommending is that you measure thyroid functions, even in people who are taking Synthroid, if you're not going to exclude them. So we debated long and hard whether to include people who are taking Synthroid or not. And we decided, because sometimes when thyroid is not functioning well, they're a little more prone to depression, that we would include them as opposed to exclude them, as long as they were receiving stable doses and everything was okay with the primary care doctor. And so out of our 60, about 8 or 10 were taking Synthroid. We expected that in our bipolar studies. We measured, and when we looked at their thyroid indices, that Synthroid wasn't altered throughout the 60 days, because many of them had been taking it for years. Interestingly, some of them had like lower thyroid function, and this thing boosted it into normal. The TSH levels came down, free T3 and T4 went up as expected, and they were normal. No one became thyrotoxic. We were very worried about that Dutch lady's experience. That didn't happen. We repeated, so we put that into the literature. There was somebody junior working with me. She wanted to write a paper. I said, this is an interesting sort of case series from the double-blind study. And she wrote it and put it into the literature. And this group from India immediately called me and said, we are very keen on your little case series because we are about to embark on a double-blind study for hypothyroidism. And they have published it and put it into the literature. So, you know, it's interesting that small, intriguing things people pick up, they're looking for it, because we've had 2 animal data sets from Dr. Panda and Dr. Carr in India showing that it boosts thyroid. A lot of medicine actually suppress thyroid, but this is one thing that actually boosts it. So what we have said to our patients is there's no absolute contraindication if you're taking Synthroid, because after all, we have studied it in our 2 small studies. But I think it's important to remain vigilant. Just keep in touch with your doctor if you were ever going to use ashwagandha, that it has this pro-thyroid effect. It could put me a little too high. So,
very good.
Yeah.
I think I take a blood test every year because of that.
Yeah.
And I will monitor it, but I will wait till this blood test, which is next month, and then start taking ashwagandha and see if the next blood test makes any difference.
Yeah.
Okay, I might add here, with the permission of Roy, that you know, the good thing of this talk today would be the practical applicability of this would be that when you are seeing your own physician for any treatment that you're having, to let them know that you are or not, or you are taking ashwagandha.
Yeah.
I don't think the purpose of this presentation today was to endorse any use of ashwagandha at all, but if you're using it, let the physician or PCB taking care of you know that you're taking ashwagandha, and they'll be aware of the side effects or not from that point of view. Okay, next person that we have is Durlabh Trivedi. He is the question.
Thank you very much.
Thank you.
I have a suggestion and a question. The suggestion is, did you contact Baba Ramdev of Patanjali? Because they conduct all these studies. They have a lot of money for research.
No, I haven't contacted him. You know,
that would be one area you should explore.
Yeah.
Because he has a lot of money and interest in this research.
Yes, definitely.
Okay.
They are very interested in Ayurvedic research, absolutely, and including ashwagandha.
Yeah, my question is that Ayurveda is a science. There are books written.
Yes.
The degree which is awarded in India.
Yeah.
Why do we need to reinvent the wheel by doing these research? It's all mentioned based on the research. For example, you do datun with neem.
Yeah. Yeah.
It's good.
Yeah.
And that neem grows here or there, it has same property. Same thing is with tulsi leaves.
Yeah.
They are known to have some curative effect. and whether tulsi plant is in my house or a house in Japan or in India, it will have the same thing.
Yeah.
So why are we doing this?
I think, like the ancient science is clear cut, so Bhushan Patwardhan, who's like a big advocate and part of Ayush and edits this journal in India. We've had some good sort of, back and forth on this. And since he's trained in this modern scientific method, he understands it from both perspectives, that plant and agriculture harvesting, historically in the unpolluted days, maybe pre-industrialization, there was less issues of where it was grown, what climate, what altitude, which season it was plucked. But over time, as knowledge has, grown, as the industrial world has polluted, and we as humans, with the dominant species on planet Earth, we have used a lot of things, and that has created consequences, including stress for plants and the plant kingdom in general. And so this has led to, even though the plant like tulsi might be grown on a pot in Pittsburgh, in California, in Japan, like you were saying, Brazil, but it could be quite different depending on soil conditions, pollution, air pollution. And this is now being shown in extracts, even crude extracts of ashwagandha. So, so for that reason, we need a little more guarantee. If something works, it'll continue working, and it's not influenced because suddenly it's cultivated in the slopes in some mountain in Hawaii, which may be hit by some volcanic ash. And, so those are some of the issues why a lot of this is being redone and re-standardized, not to deny or, make trivial what we have known for millennia.
Oh, great. Roy, thanks very much. Chetan has a question.
Yeah.
Chetan Patel.
Yes. Yes, Dr. Roy, it was a wonderful presentation. And I want more questions. I have a blood pressure as well as a cholesterol. I'm taking the 1 tablet of blood pressure and evening for cholesterol. By taking these medications, my blood pressure and cholesterol both are well in control. Do you think I should take the ashwagandha tablets? And if yes, then what should be the dose?
I think, of course, it's like Subhash was saying, you can have this conversation with your doctor, but in general, I think it's sold currently as an over-the-counter, because anyone, as long as it's safe and they can give certain labels on their product, whether it's Himalaya or any other extract, any other company, they can use it. So it's not sold as something for prescription medicine. It's not been approved for any condition, but it's sold like more over the counter, being seen as safe, as an herbal botanical. And because of general use in India and other parts of the world for millennia, it's argued that the safety to some extent has been established. But when it comes to specific illnesses, interacting, many of our people in psychiatry have hypertension, high cholesterol. They've also taken these medicines. So clearly that has happened, but the safety of it in any given individual, it's so much harder to answer. I take it on top of my cholesterol medicine, on top of my blood pressure medicine. I feel it helps in certain ways with sleep and general sort of well-being. So I use it in that way. I tell my cardiologist, I tell my family physician. Both of them don't have big issues with ashwagandha, and they may not be real experts. My family practitioner, she's very interested in Ayurvedic things, so I've lucked out. But many people in allopathic medicine may be clueless about Ayurvedic medicines. So, it's a conversation that you would have to almost research and present to your doctor in a certain way. Otherwise, they could become very nervous because they just Google some drug interaction that pops up and they say, oh my God, we don't want you dying with this product, So you have to be prepared for all kinds of personalities, even with your healthcare professional,
Right. I might make a comment here. The reason we chose this topic to present to our members and by Professor Chengappa, because ashwagandha has been used widely for many, many diseases generally, and they do not know whether it's effective or not, but hearing that it's been effective, they've been using it. So we just thought we'll provide you some science-based knowledge as much as is possible under this day and age from the right source, like Dr. Chengappa has done that thing, just for your information and knowledge. It's not for recommendation of any specific treatment as such. So I think be aware of that thing. The purpose of the whole talk is just for informational. And if you are using it, to please let your physicians know that you're using it in case there are some side effects or inter-drug interactions and all like that, that he may be aware of that thing. We have a comment from Jamnadas Thakkar. Jamnadas ji?
Yeah, namaskar.
Namaskar.
Dr. Roy, that was very good
Thank you.
talk that we enjoyed it. I want to tell you that my wife has been using ashwagandha for many, many, many years. She has acute rheumatoid arthritis. He takes 5 parts, 5 items together, equal quantity. One is ginger powder.
Yeah.
One is haldi, that is turmeric. And one is methi, what do you call this? Fenugreek. I'm sorry, I don't know the name of the.
Yeah. Fenugreek, yeah.
And ashwagandha and black sesame seeds. And every morning when she gets up with the warm water, she takes 1 teaspoon of that, and she has been very settled on her rheumatoid arthritis.
Yeah.
And she does have a thyroid also, but her thyroid and her PCP knows that, and thyroid is also well under control. Same 75 units she has been taking it, and she's been functioning without taking the actual prescription medicine for the rheumatoid arthritis. So that has good help in that. I just wanted to bring it to your attention. That does help that. Rheumatoid arthritis.
Yeah, thank you, Mr. Thakkar. I think we've had testimonials like this of people taking like a complex of herbal medicines, which is typically in Ayurveda, because whole body medicine, it's holistic. Allopathic is finally moving where Ayurveda has already been for 2000 years, and so in a way, using it in warm water at the right time in the morning with milk, with plain water, after a meal, before a meal, a lot of that very sort of careful, clever thinking is slowly coming into regular medicine, like this modern medicine. But a lot of the indigenous forms of medicine, especially Ayurveda, has been on this for a long time and has found a lot of benefits. But I've heard similar testimonials in India and even here from people, even people who don't have necessarily an Indian background, but they've had Indian neighbors, so they have experimented themselves and been very happy with the results. Yeah.
There's a question here, Roy, about the book that you mentioned about, a book by Cole.
Yeah.
Does that, or you have the book? Okay. Does that book include some general information about various diseases for the lay public as such, or is it more like scientific book?
I would say it's mostly a compilation of science. It's not so, and the absolutely last section, which is a small section, is even about growing ashwagandha, because now it's cultivated, but how to cultivate it in non-polluted environments. So the people who supply us have had a hard time getting the soil very clear in parts of Madhya Pradesh and Uttar Pradesh. So they've ended up on the border with Nepal, because that still is old soil and less industrialized soil. Yet others are into these modern methods of agriculture, hydroponics. So hydroponic agriculture they're getting into with ashwagandha plant, so that if you can get pure extracts, less contaminated, you can extract it without worrying about this and that and everything,
Right. Very well. So that book would be beneficial for the lay person to read, or not?
I don't think so. If I were to just say a yes/no answer, it's quite technical.
I see. Okay.
Yeah.
Good. And I want to remind all the members that today's meeting is being recorded and also will be available later on in case you missed it or want to see it again on Facebook also. So with that, I would request Dilip Desai to make his final comments. And Roy, we thank you. And at this time, we would have been standing up and giving you a stand up ovation as such. But since we are at home now, please accept this as a standing ovation to you. We appreciate that very, very much from you for your presence in your talk.
Thank you. Thanks. Namaste and namaskar, everyone.
And Dilip would be offer you the official thank you note now. Dilip.
Well, Dr. Roy Chengappa, I must say, myself, maybe some others too, were literally mesmerized by just looking at your slides and by listening to you. But I must say, on behalf of USAP and everyone here today, a big, big thank you for sharing your time, knowledge, experience, and giving us insights into a hot topic on one of the key Ayurvedic herbs, Ashwagandha. Your years of research and your depth of knowledge and understanding was coming out very clearly. And the way your ability to connect with the people was very impressive, which made your presentation very, very informative and helpful in grasping the potential benefits to the fullest, I would say. You highlighted the clinical trials and shared with us the promising results of each trial in treating diseases or conditions like diabetes, cholesterol, Alzheimer's, cancer, in psychiatry, etc., was not only just informative, but also very, very useful and applicable. I'm sure many will see a glimmer of hope because they or their loved ones are going through such conditions in real life. And Subhash Bhai, you did an excellent job moderating this presentation, and your pathology background was very, very helpful in trying to relate in a layman's term complicated slide, and it was very helpful in understanding. I believe many of us were naive about the subject when we walked in, and now living with a wealth of knowledge on Ashwagandha. And it is great to see so many people here because I believe they are in pursuit of lifetime learning, pursuit of living in good health, and pursuit of being happy. So USAP has offered, as we know, topics on Ayurveda in the past multiple times. But this time the topic was on a specific herb within Ayurveda, Ashwagandha, which is, I would say, one of the key powerful herbs of Ayurveda. And friends, your support and interest have resulted into another successful event for USAP. Our motto is to help you stay happy, healthy, motivated, and staying connected. So you keep on supporting and we will keep on serving. So Dr. Roy Chengappa, once again, thank you so much, very much for your outstanding presentation. People, I'm sure, really, really enjoyed. And it's really promising that so many areas of the human diseases, it has so many potential benefits coming. So thanks again, Subhash Bhai, Dr. Roy Chengappa. And I move over back to you, Subhash Bhai.
Yeah, thank you very much, Dilip. And Roy, one of the missions of USAP is to promote healthy aging.
Yeah.
And I think by your presentation, you helped us reach that goal today.
Thank you.
We again thank you very much, and we look forward to your support and help in the future also.
All right, thank you.
Yeah.
I just want to mention to you members that Roy Chengappa has been a conductor and moderator for Psychiatry Journal Club once a month for the last 20 years.
Wow.
Yeah, that's a Raj Lal speaking, a psychiatrist, a fellow psychiatrist, and she knows Roy like nobody else does. So thanks, thanks, Raj.
Thank you. No, I mean, we like moderator and, you know.
Yes.
And
I would like to say,
Suman was a big part of that, you know.
Yes, my wife Suman also was an associate with Roy, also in their journal clubs, also for a long time. Also, thanks, Roy.
Yeah.
Subhash bhai, one more thing I would like to add is we have seen some few comments, positive feedback for our esteemed guest or the speaker today. We still have a few more minutes, so if you would like to add some more comments, please do so, and we can use some of those comments in our newsletter. And it's always nice to see some positive feedback, either on the speaker, either on the quality of the presentation, or on the USAP. And if I would say, even if you have any other suggestion for USAP to consider, any other topic in future that we should bring, please add that in your comments. So we will definitely consider that in our future presentations. Thank you.
Very nice. So with that, then I take your leave, Roy, and take leave from everybody else. And we will end the meeting today. I thank you all very much for attending this meeting today. Thank you.
All right. Thank you, everyone.
Thank you.
Dr. Roy, we got a comment that they loved your Desi outfit.
Yeah. I was telling Subhash, I was telling my wife, I think with Ganesh Chaturthi and USAP, I should have a totally Indian outfit.
You look very nice. Thank you.
Thanks. Bye-bye.
Bye.
Bye. Jai Shri Krishna. Thanks.
Okay.
Hi, Salek Roy.
Thank you all. So
thank
let's see. How many people we have left now? 33 people are still there.
Double three.
It was an awesome talk.
Yes,
Without any
definitely.
question.
I have one question. This is Pramila.
I think the speaker may have left already, but go ahead.
No, not for him. I had it. I raised my hand, but I think Subhash Bhai didn't see me.
Oh, I'm sorry. Okay.
Because I have a really good question, but so it doesn't matter next time. But I don't know why he couldn't see me because I was raising my few times.
Oh,
okay. I didn't see any ladies have raised
Maybe you have to put in a comment. Maybe you have to put in a chat box.
We can help you.
Put any comment. This is just a.
If you want to send Subhash Bhai and ask that question so he can get it. Looks like he's the closest to Dr. Roy.
Yes. Okay. That's all I want to tell.
Okay. All right. Chetan, anything else?
Nothing to do with
Chetan Bhai, anything else you want to add?
Yes, Premila ji, if you question, just email to the Subhash Bhai and we will definitely try to get the answer of your question. And please mark your calendar for the September 20. We have wonderful presentation on the finance management and more information will come soon. So please mark your calendar. You will learn a lot of information of the financial management. That is September 20. And for the golf flower, we have golf outing on the September 16. That is Wednesday at 11 o'clock. So those who are interested, please give your name to Sudhir Patel or Jayant Mirani.
Okay, so I'm going to save all my chat and then we close the meeting. Okay.
Thank you. Thank you.
Thank you all. And
very nice.
Very good.